Popular culture made dinosaur cloning feel almost inevitable, but the real biological pathway breaks at nearly every step. The famous proposal begins with a mosquito trapped in resin after feeding on a dinosaur. That image compresses collection, preservation, sequencing, reconstruction, cloning, and development into one deceptively simple gesture. Reviving an extinct animal would require an almost complete, correctly ordered genetic instruction set, rather than a trace molecule. The central obstacle is deep time. Chemical information does not survive as long as fossil shapes can. After death, water, heat, oxidation, microbes, and radiation continually damage genetic material. Fossilization can preserve anatomy while replacing, altering, or destroying much of the original biological material. A beautifully preserved skeleton can reveal the body's structure without retaining a readable genome. Amber can preserve astonishing visual detail, yet appearance alone says nothing about intact dinosaur DNA. Even before sequencing begins, modern contamination can overwhelm any genuine ancient signal. Ancient remains can preserve several kinds of evidence, but those categories must not be confused.
Recently frozen animals are fundamentally different from dinosaurs that vanished tens of millions of years earlier.
De-extinction becomes more plausible when remains are young, cold, and closely related to living species. Short molecular scraps cannot substitute for chromosomes containing millions of ordered instructions. Extremely small signals are difficult to authenticate because laboratory and environmental DNA are everywhere. Credible ancient DNA research depends on contamination controls, reproducible results, and molecular damage patterns. Proteins may outlast DNA and still reveal ancestry, tissue type, or aspects of physiology. Useful proteins can enrich a reconstruction without supplying the instructions required to build the organism. Pigment structures can sometimes suggest coloration, yet they offer no path toward cloning. Exceptional fossils can preserve surprising chemistry, but extraordinary preservation still falls far short of a viable cell. Cloning starts with organized genetic material inside a functioning cell, not merely chemical traces. A genome includes genes, control regions, chromosome structure, and developmental timing information. Development depends on when genes activate, where they act, and how their signals interact. Even numerous fragments would leave enormous unknown regions that cannot be guessed safely. Rebuilding a dinosaur genome would resemble restoring a vast book from scattered letters and a distant relative's edition. Bird genomes provide evolutionary clues, not a complete template for any lost non-avian species. Shared ancestry does not erase millions of years of genomic change, specialization, and extinction. Evolution changes sequences, gene regulation, chromosome organization, and developmental pathways across deep time. A reconstructed sequence must operate correctly from the first division through every developmental stage. Guessing across those uncertainties would more likely produce failed development than a living dinosaur. Standard cloning transfers an intact donor nucleus into an egg whose own nucleus has been removed. The procedure requires living chromosomes, functional cellular machinery, and compatibility with the egg environment. Cloning remains inefficient even when donor and surrogate belong to closely related living animals. No dinosaur fossil provides the intact living nucleus that nuclear transfer cloning demands. Scientists also lack a known egg environment capable of supporting a non-avian dinosaur embryo. An egg is a complex physiological system, rather than a passive container for DNA. Maternal molecules, nutrients, immune interactions, and timing would all need to match an extinct embryo. Living birds are dinosaur relatives, yet no modern species is an interchangeable surrogate for every extinct branch. Artificial incubation would still require detailed developmental natal knowledge that fossils cannot provide. The cloning route fails through several independent barriers before an embryo could ever exist. Birds offer a different research path because they are living dinosaurs with working cells and genomes. Developmental biology can test whether modern birds retain pathways related to ancestral dinosaur traits. Changing tail development could produce a bird with a more ancestral-looking profile. Researchers can investigate tooth-related pathways, but reactivating fragments of development does not restore an extinct jaw system. Skull shape emerges from interacting tissues, genes, growth rates, and mechanical forces. Wings retain ancestral bones, yet evolution has reorganized their proportions, function, and development. An engineered bird might resemble a dinosaur in selected features while remaining a modified avian organism. Clear language matters, because resemblance, ancestry, cloning, and restoration describe different biological outcomes. Future biotechnology may create proxies or ancestral-looking traits without recreating an original extinct species. The plausible experiment starts from living avian biology. It does not recover a lost dinosaur genome. Extinction projects differ enormously because age, tissue preservation, genome quality, and living relatives vary. Mammoth research benefits from recent remains and close living relatives, yet even that work aims at approximation. Recently lost species may leave museum tissues and clearer genomes that dinosaurs never provided. Some projects seek ecological proxies by editing close relatives, not by recovering an original animal cell. Non-avian dinosaurs combine extreme age with missing cells, incomplete molecules, and distant living references. A species is embedded in an ecological network that also disappears or changes after extinction. Microbial partners influence digestion, immunity, growth, and behavior. Yet dinosaur microbiomes are inaccessible. A recreated proxy could encounter modern pathogens and immune challenges its extinct counterpart never faced.
Technical possibility would not answer whether creating a contextless, vulnerable animal was ethically justified.
Resources spent chasing cinematic resurrection must be weighed against research and conservation with clearer benefits. Paleontology already reconstructs dinosaur biology through multiple independent lines of evidence. Imaging can reveal internal anatomy without just destroying the fossil. Skull structures help estimate how extinct animals sensed and moved through their environments. Microscopic bone structure records growth rate, maturity, injury, and physiology. Chemical signatures in teeth and bones can preserve clues about diet and habitat. Biomechanics tests how jaws, limbs, and skeletons could function under physical constraints. Trackways capture movement and social patterns that skeletons alone cannot show. Nests and embryos reveal reproduction, parental care, and developmental strategies. Feathers preserve evidence for insulation, display, flight, and sometimes coloration. The most informative living dinosaur laboratory is the diversity of birds themselves. Bird anatomy preserves numerous traits inherited and transformed from theropod ancestors. Shared bones reveal continuity far more directly than any speculative clone. Evolution altered the body gradually, while preserving a recognizable structural inheritance. The asteroid ended most dinosaur lineages, but one avian branch crossed the boundary. Several traits may have improved survival during the ecological collapse after impact. Access to persistent food could have mattered when plant-based ecosystems failed. Smaller bodies and shorter generations can help populations endure and rebound after catastrophe. After the extinction, surviving birds diversified into ecological roles left vacant by vanished groups. Living dinosaurs now occupy streets, farms, forests, oceans, and skies around the planet.
The genuine dinosaur connection is visible in living birds, not hidden as a complete genome inside amber.
The cinematic shortcut works because it hides the number of assumptions between specimen and animal.
Every hidden checkpoint requires evidence that does not exist.
A mosquito's meal is processed biologically rather than stored as a pristine vial. Resin chemistry and later contamination further weaken the idea of recovering clean dinosaur blood. Ancient molecular claims survive only when independent tests exclude modern contamination. Unexpected structures inside fossils do not automatically contain viable cells or DNA. Storytelling needs a clean mechanism, while science must confront every missing piece. The idea persists because it combines powerful animals, lost worlds, and biotechnology in one memorable image. The real story becomes clearer when evidence is ranked by what it can genuinely support. Exact cloning is not credible. Limited trait engineering is conceivable. Dinosaur research is already highly productive. Biotechnology will advance, but progress will not erase the information destroyed by deep time. More sensitive tools can detect smaller traces without creating molecules that no longer exist. Synthetic biology could build an engineered genome, but that product would reflect modern choices and guesses. Future experiments may create birds with unusual ancestral-looking features, while remaining avian by ancestry and design. Biological identity depends on far more than a few visible characteristics. Even an engineered proxy would inhabit a modern ecosystem assembled from imperfect substitutes. Creating an animal also creates responsibilities that a dramatic laboratory montage cannot answer. The closest thing to saving dinosaurs is protecting the avian dinosaurs still alive. Modern birds offer an extraordinary range of dinosaur evolution that requires no resurrection fantasy. The complete answer emerges by following each piece in the correct biological order. Amber preserves form beautifully, but it has not yielded a complete dinosaur genome. DNA degrades across time, leaving dinosaur-age material far beyond known cloning requirements. Fossils provide no intact living donor cell for nuclear transfer. No known egg or surrogate can be assumed compatible with an extinct non-avian embryo. An original species also depended on ecological relationships that cannot be restored precisely. Birds preserve the only living continuation of dinosaur evolution. Four independent barriers make exact dinosaur cloning scientifically untenable. Realistic science lies in reconstruction, evolutionary biology, and carefully defined genetic experiments. The evidence offers a richer story than resurrection, transformation, survival, and deep evolutionary continuity. The strongest questions ask how dinosaurs lived, changed, and survived through birds. The dinosaur outside your window is scientifically more authentic than any fictional clone. The story begins with Amber, but the evidence ends with evolution. One dinosaur lineage survived the extinction and continued changing into the birds alive today.
The materials needed for cloning disappeared, while the evolutionary lineage itself endured.
Science cannot reproduce the cinematic pathway, but it can explain why that pathway fails. Conclusion does not depend on a single missing technology. It follows from lost information, incompatible development, and deep evolutionary change. Fossils cannot become living cells, yet they can reveal extinct lives with remarkable precision. The living legacy of dinosaurs is not locked inside a specimen. It is moving through modern ecosystems. Cloning a dinosaur remains beyond credible science, but understanding dinosaurs has never been more powerful. Jurassic Park remains unforgettable fiction. The real dinosaur story survives in fossils, evolution, and every bird still flying. The dinosaurs did not vanish in one explosion. Their extinction began with 24 hours that permanently rewired the planet. Before impact, life occupied nearly every habitat. None of it could sense the mountain already approaching from space. The object was probably a carbon-rich asteroid, roughly 10 to 15 kilometers wide and moving at extraordinary speed. Its size was terrifying. But size was only half the weapon. The other half was velocity. It crossed roughly 20 kilometers every second. At that speed, an ocean becomes almost irrelevant. Its destination was a shallow tropical sea near Yucatan, a target that amplified the catastrophe. Below the sea lay carbonate and sulfur-bearing rocks, materials capable of turning impact energy into climate-changing dust and gases. Shocked crystals, glass droplets, crater rocks, and global sediments preserve the event, like damaged frames from a lost film. There was no warning system, no evacuation, and nowhere on Earth prepared for what was seconds away. The atmosphere barely slowed it. Air compressed ahead of the asteroid into a descending wall of incandescent plasma. Near Yucatan, the horizon brightened beyond sunrise. Animals had seconds between noticing the light and being overtaken by it. From the upper atmosphere to the surface took less time than a breath. Then the Cretaceous world changed forever. Contact released energy near 10 to the 23rd joules, far beyond every weapon humans have detonated. The asteroid did not remain intact. It and the seafloor were crushed, melted, and vaporized almost instantly. Crater geometry suggests a steep, oblique strike, an angle efficient at launching vaporized target rock into the atmosphere. Ordinary minerals became extraordinary evidence. Quartz deformed, sediment became glass, and ancient seabed entered the sky. Within the nearest region, survival was effectively impossible. Heat, pressure, debris, and wind arrived together. The explosion remained regional for only moments. Its fastest material was already climbing high enough to become a global disaster. The impact excavated a temporary cavity far deeper than the crater that would remain. Earth's crust opened like a wound. Before the crater finished forming, seismic energy was already racing through the body of the planet. Compressed crust rebounded upward. For a moment, solid rock rose like liquid inside a planetary splash. Gravity pulled the central uplift apart, spreading deep crust outward into Chicxulub's distinctive peak ring. Within minutes, the collapsing structure produced a crater nearly 200 kilometers across. The visible crater was only the surface expression. Beneath it, the crust was shattered, heated, displaced, and made permeable. An ejecta curtain expanded outward. Large fragments fell nearby, while faster dust and droplets began crossing continents. Near Yucatan, enormous blocks returned first, striking land and sea while the main blast still expanded. Higher in the plume, vaporized rock cooled into glass droplets that became a worldwide impact signature. The sea had been thrown outward. Gravity now drew a chaotic wall of water and debris back toward ground zero. Returning water collided with falling ejecta and collapsing rock, building a thick geological record within hours. Scientists later drilled this deposit and found the first day of the Cenozoic preserved inside stone. A supersonic pressure wave raced outward, turning the atmosphere itself into a moving instrument of destruction. Many forests were flattened before they burned, stripped by winds stronger than the most violent storms. Thermal radiation reached exposed surfaces before the slower pressure wave and much of the falling debris. Near the crater, vegetation could ignite quickly. Farther away, fire depended on fuel, shelter, weather, and distance. The collision generated seismic shaking beyond ordinary earthquakes, sending waves through crust, oceans, and continental margins. Thousands of kilometers away, the ground could move before animals saw any debris from the impact. Shaking destabilized underwater slopes, triggering landslides that buried seafloor ecosystems beneath moving clouds of sediment. Even enclosed lakes and inland seas could oscillate violently as seismic energy crossed entire continental plates. Along some coasts, water withdrew first. What looked like escape was actually the warning of a much larger wave. The first regional waves carried trees, sediment, rock, and marine debris far beyond the former shoreline. The impact displaced an ocean. Its energy divided among coastlines, deep basins, narrow passages, and continental shelves. Models estimate vastly more tsunami energy than any historically recorded event, an ocean-scale disturbance without human comparison. In deep water, the tsunami might not resemble a towering wall. Its danger was hidden in speed and moving mass. As the tsunami entered shallow water, it slowed, compressed, and grew, concentrating distant impact energy against coastlines. Many coasts were struck repeatedly. Waves arrived, withdrew, and returned, rebuilding the shoreline after every surge. Powerful currents disturbed seafloor sediment, leaving geological traces even far from the crater. Impact-driven surges could travel inland and upstream, mixing marine debris with freshwater landscapes within minutes. Some distant deposits may record water surges occurring while impact glass was still falling. There was no single disaster front. Blast, earthquake, ejecta, and tsunami arrived in different orders. Within the first hour, one impact had become thousands of local catastrophes, shaped by geography and distance. The fastest ejecta now began returning from thousands of ballistic trajectories around the planet. Each returning fragment heated the atmosphere. Multiplied by immense quantities, that energy became a planetary concern. Across parts of Earth, the sky may have resembled a meteor storm generated by the planet itself. Re-entry heating varied with clouds, topography, water, and shelter. Exposure could determine immediate survival. Scientists debate how globally the heat ignited forests. Evidence supports widespread burning, not necessarily one simultaneous fire. Where forests burned, fire added soot to air, already overloaded with pulverized rock and impact gases. Shelter suddenly mattered more than strength. Water, soil, and small body size could buffer the first thermal assault. Open ground, forest canopies, and coastlines combined exposure to heat, debris, fire, and violent water. Cooling glass spherules fell worldwide, creating a thin time marker later recognized in rock. The first hours ended with a brutal reversal. After unbearable brightness, Earth began losing the sun. Vast atmospheric currents carried impact material around Earth. Within hours, the crisis crossed hemispheres. Heavy debris fell quickly. Fine dust, soot, and aerosols remained aloft and began controlling climate. As the atmospheric veil thickened, less sunlight reached the surface. Impact winter was beginning to assemble. Sulfur from the target rocks began forming reflective aerosols, although its total quantity remains debated. Fine silicate dust may have darkened Earth long enough to suppress photosynthesis on land and at sea. Soot added another dark layer, potentially intensifying the developing impact winter. Far from Yucatan, surviving animals faced a new threat: daylight fading without any local disaster. Photosynthesis did not stop everywhere instantly, but the energy entering food webs had already begun to fall. During the first day, darkness advanced faster than the full temperature collapse. Normal night crossed Earth. Behind it came a darkness that would not end with sunrise. While the atmosphere transformed above, the tsunami continued carrying impact energy through the Atlantic basin. Hours after impact, coastlines far from Mexico could still be struck by waves from an event they never witnessed. Continents blocked some paths and focused others. Distance alone did not determine tsunami danger. Around islands and peninsulas, waves bent, reflected, and interfered, producing locally extreme conditions. Even where coastlines escaped the highest water, deep currents could scour sediment and disturb seafloor habitats. The tsunami struck ecological bottlenecks: reefs, estuaries, nesting grounds, and coastal forests.
Marine ecosystems were physically damaged before climate effects fully arrived: turbulence, burial, and lost shallow water habitat.
The waves did not disappear after landfall. Reflections kept ocean currents disturbed long after the first arrivals. Survival now depended on location. Different habitats faced water, fire, debris, darkness, or combinations of all four. Where water receded, it left salt, sediment, debris, and buried food across landscapes that no longer resembled home. On Earth's far side, many large animals may still have been alive. Their world was already changing overhead. The first clues could be subtle: haze, falling grit, weakened light, unusual behavior, and spreading silence. Burrowing, aquatic habits, flexible diets, and small body size suddenly became powerful survival advantages. Food webs based on seeds, fungi, carrion, and detritus had temporary reserves when fresh plants failed. Freshwater systems offered shelter, stored organic matter, and temporary protection from heat, fire, and starvation. Large dinosaurs required abundant food, space, stable reproduction, and functioning ecosystems, exactly what the impact dismantled. Evidence suggests Northern Hemisphere spring, potentially striking many organisms during growth and reproduction. Ocean plankton still floated beneath the surface, but the sunlight supporting them was being filtered away. For some regions, the next sunrise arrived weak and distorted. Elsewhere, ordinary night simply deepened. Half a day after impact, Earth still rotated normally. Almost every surface system was behaving abnormally. planetary winds transformed a directional blast into an increasingly global atmospheric crisis. Heat, vapor, ash, smoke, and pressure disturbances created ingredients for violent, unfamiliar weather. Acidic precipitation added stress, although its timing and intensity varied across the planet. Local heat could persist even while Earth's broader energy balance turned toward rapid cooling. Every hour of reduced sunlight weakened primary production, beginning invisibly inside photosynthetic cells. Large herbivores did not need to die immediately. They only needed plants to stop recovering. Predators were one step farther from impact, but only one broken food link from starvation.
Impact gases changed seawater chemistry, adding chemical stress to darkness, turbulence, and disrupted food chains.
Extinction was selective, not random. Biology mattered, but shelter, diet, geography, and chance mattered too. Earth completed most of a rotation beneath the impact cloud. The new sky could persist for years. The first day did not kill everything. Pockets of life remained in water, soil, debris, and dormant tissues. Some dinosaurs probably survived the first day. Immediate survival was not long-term escape. For large animals, every remaining resource was finite: food, water, shelter, nesting space, and prey. Smaller survivors needed less food, hid more effectively, and used resources large specialists ignored. Seeds, spores, roots, and underground tissues could wait, while exposed forests could not. Deepwater organisms escaped fire and blast, but many still depended on food produced near the sunlit surface. As primary producers declined, extinction pressure climbed from microscopic cells toward the largest marine predators.
Flexible ecosystems could bend. Specialized ecosystems broke when one essential relationship disappeared.
By the 18th hour, spectacular violence was easing in many places. The quieter catastrophe was taking control. The disaster was changing from impact fight physics measured in seconds to ecological collapse measured in years. At ground zero, Chicxulub remained a furnace of fractured rock, hot water, debris, and unstable walls. Layer by layer, the crater recorded its first day. Melt below, chaotic debris above, then resurge sediment. Charcoal in the crater shows burning occurred quickly enough for fire debris to arrive during early sedimentation. The crater contains little sulfur-rich target rock, suggesting much of it escaped into the atmosphere. Worldwide, the same moment became a thin boundary containing impact glass, shocked minerals, soot, and extraterrestrial metal. After 24 hours, Earth still existed. Many organisms still breathed, but the old planet was already gone. The asteroid did not need to kill every dinosaur that day. It only needed to destroy their life support systems.
The decisive extinction unfolded later as darkness, cooling, chemical disruption, and starvation persisted.
One world dominated by non-avian dinosaurs was closing. Another was beginning among unlikely survivors. The first 24 hours did not complete the extinction. They created a world the dinosaurs could not rebuild. Beneath every ocean, continent, and city, Earth may be storing an astonishing amount of water hundreds of kilometers underground. But this is not a liquid ocean in a cavern. The water is chemically locked inside solid minerals. At these depths, water exists mainly as hydrogen bound into crystal structures, not as free-flowing lakes or seas.
The most important zone lies roughly 410 to 660 kilometers below the surface.
Geologists call it the mantle transition zone, a pressure-built layer between the upper and lower mantle. This hidden reservoir matters because Earth's surface ocean and deep interior are part of one extremely slow water cycle. Water can travel downward with sinking tectonic plates, disappear into minerals, and return through melting and volcanoes. The difficulty is obvious. No drill can reach anywhere close to the transition zone. So scientists investigate this water indirectly, using seismic waves, laboratory pressure experiments, and rare minerals carried upward from depth. Then one tiny diamond inclusion provided the first direct sample from this hidden reservoir. To understand that discovery, first strip Earth into the layers pressure creates beneath our feet. The shallow upper mantle is dominated by olivine, a mineral with relatively limited capacity to store water. As pressure rises with depth, the same chemical ingredients rearrange into denser crystal structures. Near 410 kilometers, olivine transforms into wadsleyite, a mineral that can hold far more water. Wadsleyite does not contain underground droplets. It stores hydrogen inside defects in its crystal lattice. Deeper still, wadsleyite changes into ringwoodite, another high-pressure mineral with major water storage capacity. At about 660 kilometers, ringwoodite breaks down into minerals that generally hold much less water. That sharp drop in storage capacity can force water out and help create tiny amounts of melt. The transition zone may therefore act like a deep sponge, and, at its boundaries, like a water trap. But a mineral's capacity to store water does not prove the entire global layer is filled to that capacity. Because the transition zone is unreachable, scientists read its properties through the way seismic waves travel. Abrupt mineral changes at 410 and 660 kilometers create seismic discontinuities that geophysicists can detect. Seismic tomography converts thousands of travel times into images of faster, slower, colder, and hotter mantle. Their depth, sharpness, and local variations reveal temperature, composition, and possibly the presence of water or melt. Some low-velocity layers near the transition zone boundaries are consistent with small amounts of partial melt. Laboratories test that interpretation by recreating deep Earth pressures and temperatures in diamond anvil cells. When hydrous ringwoodite transforms under lower mantle conditions, experiments can produce melt between the new mineral grains.
Seismic observations beneath North America found velocity drops where mantle flow models also predicted downward movement across 660 kilometers.
Together, experiments and seismic data support dehydration melting as one way water may remain trapped near the transition zone. Still, seismic signals and experiments were indirect. Scientists needed a piece of the deep mantle itself. The crucial sample came from a rough diamond recovered near Juína, Brazil. Deep source eruptions had carried the diamond upward fast enough to preserve a mineral formed hundreds of kilometers below. Inside the diamond was a microscopic inclusion, too small to see without specialized instruments. X-ray diffraction identified the inclusion as ringwoodite, the mineral expected in the lower part of the transition zone. It was the first reported terrestrial occurrence of this high-pressure olivine polymine polymorph in a natural sample. Infrared spectroscopy showed that the inclusion was water-rich, in the mineralogical sense. It contained substantial hydroxyl. The preserved inclusion indicated roughly 1 weight% water, locally, within the transition zone mineral. That was direct evidence that at least part of the transition zone is hydrous, not proof that the whole layer is equally wet. The diamond solved one question and sharpened another. How representative was that tiny piece of deep earth? A speck inside a diamond had transformed the debate from pure theory into direct observation. The discovery was widely described as an ocean inside Earth. But that phrase can be deeply misleading. The mineral stores water as hydroxyl, hydrogen bonded within the crystal, not liquid water filling open space. Hydrogen can substitute into defects where metal ions would otherwise occupy the structure. Each grain stores only a small fraction by mass, but the transition zone contains an enormous volume of rock. Across a planetary shell, even 1% water would represent a quantity comparable to vast surface reservoirs. Experiments show wadsleyite and ringwoodite can hold several percent water by weight under suitable conditions. If the entire zone were near saturation, its capacity could equal several surface oceans, but that is a hypothetical upper limit. The real mantle is almost certainly uneven. Wetter near some subduction systems, drier beneath other regions. Where tectonic plates carry hydrated rock deep into the mantle, local transition zone water may become concentrated. Temperature, iron content, mineral chemistry, and mantle flow all change how much water these crystals can hold. Water changes electrical conductivity, seismic speed, strength, and the way mantle rock deforms. Even when hidden inside crystals, hydrogen can make mantle minerals easier to deform over geologic time. That means deep water can influence mantle convection and the long-term movement of tectonic plates. The deep cycle begins when surface water reacts with oceanic crust and becomes stored in hydrated minerals. Sediments, altered crust, and hydrated mantle rocks can transport water toward a subduction zone. As the plate sinks, rising heat and pressure destabilize many water-bearing minerals. Much of that water escapes at shallower depths, helping generate magma beneath volcanic arcs. Volcanoes return part of the subducted water to the surface through magma and gas. But not all water returns immediately. Some hydrated material survives and continues much deeper. Inside the transition zone, wadsleyite and ringwoodite provide new crystal structures capable of storing that hydrogen. The result may be localized reservoirs shaped by ancient slabs, mantle circulation, and mineral reactions. When hydrated material crosses a boundary into minerals with lower storage capacity, water must move somewhere else. Some enters melt, some may migrate upward, and some can be recycled back into the transition zone. This is the deep water cycle, a circulation measured not in days, but in millions of years. Its pace is almost unimaginable, yet over Earth history, it can move enormous quantities between surface and interior. At the base of the transition zone, ringwoodite transforms into bridgemanite and ferropericlase. Because these minerals hold less water, the transformation can trigger dehydration melting. A 2014 study combined high-pressure experiments, seismic imaging, and mantle flow models to test this process. The observed pattern was consistent with partial melt forming where hydrated transition zone material moved downward. Even a very small melt fraction can strongly reduce seismic velocities and change how the boundary appears. If melt rises back upward, dehydration melting could help keep water concentrated inside the transition zone. Together, melting at both boundaries may create a long-lived geochemical filter around the deep reservoir. A similar release may occur when hydrated wadsleyite rises above 410 kilometers into shallower olivine stability. This does not prove a continuous global ocean. It describes mineral-bound water, possible melt, and a highly variable mantle. Every estimate of deep water depends on combining evidence that sees the mantle in a different way. Electrical conductivity can increase when hydrogen is present, but temperature, melt, and composition can produce similar signals. Seismic data also require interpretation. Slow waves may indicate water, melt, documentaries, high temperature, or chemical differences. The mantle is heterogeneous, so one region cannot automatically represent the entire planet. The diamond is direct evidence, but it is still one tiny sample from one locality. Even the diamond's source history depends on models of deep eruption, mantle flow, and mineral preservation. As a result, published estimates of the transition zone's total water content still span a wide range. New deep diamonds, better experiments, and denser seismic networks can gradually narrow that uncertainty. Future maps may reveal whether deep water forms broad reservoirs, narrow slab-fed zones, or a constantly shifting patchwork. The answer will probably be dynamic, because mantle water moves with a planet that never stops convecting. Water weakens mantle minerals, changes viscosity, and can redirect how heat and material circulate. That links deep water to plate tectonics, mantle mixing, and the generation of magma. Water helps rock melt at lower temperatures, making it a powerful ingredient in magma production. Volcanic degassing returns deep water to the atmosphere, where it can eventually rejoin rivers and oceans. Surface water then enters new crust, rides tectonic plates, and begins the journey downward again. The surface ocean is visible and fast-moving. The deep reservoir is hidden and geologically slow. Over billions of years, Grevenst exchange between them may help shape the long-term stability of Earth's water budget. Earth's oceans did not evolve separately from the mantle. The planet's interior has always been part of their history. A planet's mineralogy and deep water cycle may help determine whether water stays at the surface, enters the interior, or escapes. For Earth, the transition zone may be a bottleneck that stores water and controls how easily it crosses into deeper layers. Its mineral phase turns pressure boundaries into valves in the deep water cycle. The story is not an underground sea. It is a planet-sized chemical circulation through solid rock. Its central reservoir lies in the mantle transition zone, between 410 and 660 kilometers deep. Wadsleyite and ringwoodite can store hydrogen in crystal defects at concentrations far above shallow mantle olivine. A diamond from Brazil provided direct proof that at least one part of this zone held substantial water. But one local sample cannot tell us how wet the entire transition zone is. Scientists therefore combine diamonds, seismic waves, conductivity, experiments, and models to estimate the hidden inventory. The minerals may have capacity for several ocean equivalents, while the actual amount remains uncertain and uneven. What is certain is the cycle. Water moves down, changes minerals, and melting, and eventually returns toward the surface.
Even chemically bound water can influence the mechanics of the planet above it.
So the phrase "ocean inside Earth" is useful only if we remember what it really means. One reservoir flows in waves. The other is stored atom by atom inside rock. The next discoveries will come from smaller samples, better instruments, and clearer images of a world we cannot visit. The biggest question remains simple. How much water is actually moving through Earth's hidden mineral reservoir? The answer will reshape how we understand oceans, volcanoes, plate tectonics, and the long-term evolution of the planet. Earth is not hiding a sea beneath our feet. It is hiding water inside the architecture of the planet itself. That line connects rain, oceans, sinking plates, deep minerals, magma, and volcanoes across geological time. The familiar water cycle runs through clouds and rivers. The deeper cycle runs through rocks and tectonic plates. Deep storage can last far longer than any surface reservoir, even though the mantle itself keeps moving. Every journey through the mantle changes the mineral container and therefore changes where the hydrogen can go. That is why pressure, not empty space, creates Earth's hidden water reservoir. The reservoir is invisible because the rock still looks like rock. The water exists in the chemistry. A microscopic inclusion revealed a process operating across a layer thousands of kilometers wide. We know the mantle can hold oceans of water. We still do not know exactly how much it holds today. What lies beneath our feet is not a secret ocean. It is a hidden planetary cycle written into minerals. And that hidden reservoir may be one reason Earth has remained a water world for billions of years.